Again, let's go back here and look at the structure. It's important to note that A, a purine, pairs with T, a thymine. So that means we only have 3 rings across the two double strands, or across the 2 strands, making a double strand. Notice C is a pyrimidine and G, guanine, is a purine. Again, that's 3 base pairs. Again, that means that as we progress down our double-stranded DNA within the side chains, we will have our bases represented by the letters, and we will always only have 3 bases spanning. So that means we won't have part of our double-stranded DNA closer together with only 2 rings and further apart with 4 rings. We would see the 2 rings of 2 pyrimidines bound together and the 4 rings of 2 purines bound together. So this is the basic structure of DNA. This backbone, represented by this line, are sugar phosphates alternating, as is the same here, sugar phosphates alternating. And then these are our nitrogenous bases. Again, a purine always pairing with a pyrimidine. So how do we replicate our DNA? How do we get new DNA from old or template DNA? So we're going to highlight 4 basic steps. Here's our very, very small section of a double-stranded DNA molecule. Again, here's our sugar phosphate backbone and our sugar phosphate backbone. These are our nitrogenous bases, and they're held together by hydrogen bonds, which we learned previously are individually weak but collectively strong. So we basically have to use our original DNA as a template for our new DNA. And in these illustrations, our original DNA are represented in blue and our new DNA are represented by this pinky red color. What separates our old DNA is separating the hydrogen bonds that are holding our complementary base pairs together. And the enzyme that does that is DNA helicase. So it basically unwinds or separates our complementary strands. Not along the entire DNA molecule, but along the section that it's replicating, which is called a replication fork. As those are separated, there's an enzyme called DNA polymerase that reads the original strand and enables a complementary nucleotide to be assembled against the original strand. So for example, here we have guanine, so our DNA polymerase molecule would enable complementary base, which is cytosine, to be laid down. And here, when our DNA polymerase reads the thymine, and adenine, which is complementary, would be laid down, and so forth. So we end up following our DNA base pair rules, where guanine pairs with cytosine and adenine pairs with thymine. Once we've assembled those bases by the action of the enzyme DNA polymerase, the enzyme DNA ligase comes in and seals up our sugar phosphate backbones. So now our end result is 2 double-stranded DNA molecules, but now each double-stranded DNA molecule is made up of half original and half new. So we do have 2 strands, but one strand came from the original, and the other strand, the complementary strand that was just assembled, was assembled from nucleotides found in the nucleus. This is called semi-conservative replication because we are conserving half of the original molecule in each of our new molecules. And this helps cut down on errors in DNA replication because we're carrying our pattern with us. This is an illustration showing you a replication fork, which is the area where replication is occurring. Notice up here we have not replicated any of our DNA, so that means our DNA replication procedure is occurring in this upward direction. Here we can see we've separated our complementary bases in our original DNA. Here we can see we are laying down our new DNA, And we're laying it down on the section to the right in pieces, whereas on the left we're laying it down in one whole piece. And that's because DNA runs in two different directions. It has a direction called a 3' to 5' end. There would be a 5' up here. These are the orientations of the carbon sugars, or excuse me, the carbon atoms in the sugar molecules. And here we have our orientation from 5 to 3. So they're basically running in opposite directions. So that means the replication process on the continuous side can occur continuously because the polymerase enzyme reads in a 3' to 5' direction, so it can just basically hop on and just read down this original strand. Whereas here it can't. It has to read a section, because it has to read in a 3' to 5' direction, and the 5' end is the free end on this side. So it's going to read in pieces. Those are called Okazaki fragments that are formed. And these fragments that are formed then have to be sealed together by the enzyme DNA ligase. So here we can see the DNA polymerase molecule working in discontinuous replication. It hasn't read to lay down these complementary base pairs yet. Here it's finished this piece already and it's finished this piece already, but we see there's a gap in the bonding of the sugar phosphate backbones, so that's where the enzyme DNA ligase will come into play. Our end result, as we said, from one parent strand are two new strands, but each new strand is bound to the original parent strand. So in total, we have now two double-stranded DNA molecules, but each double-stranded DNA molecule is made up of one old strand and one new strand. Again, that's what we term semi-conservative replication. So let's move to the final part of our presentation, which is called somatic cell nuclear transfer, which is a process of cloning. And somatic means body, basically means any cell in the body, like a body cell, except for a gamete or a sex cell. And in this cloning process, you're going to transfer the genetic material from a body cell, which is diploid, to an egg cell after the egg cell has had its haploid genetic material removed. That means that egg cell is not going to need the other half of its genetic material from the father because it's already a diploid cell that you're putting in. So you're going to end up with an individual identical or cloned from the parent, so it will be identical to the parent. So this is the process that was used to produce the cow that we'll see on the last slide from its parent cow, which it was cloned from. So the two cows' names are Liz, and her clone was Liz 2. So the cow was cloned through somatic cell nuclear transfer, and these are the steps. So in this first step, we can see the cow egg. This is the cow egg. And we can also see, because of staining, the genetic material of that cow egg. And we see a micropipette over here coming in the screen that's going to remove that nuclear material. So the nuclear material has been removed from the cow egg. And all that we have in here is the cytoplasm and the cytoplasmic organelles. They've removed all the genetic material from the cow egg. Now we can see a new micropipette coming into the picture, and it's going to supply to this egg that has had the genetic material removed a donor cell. So that's what we see over here. Here we can see the donor cell being added, and this donor cell is a body cell or a soma cell. So this is where the somatic cell transfer piece comes in, and the genetic material in this donor cell is diploid. You can see the pipette is being withdrawn, And inside of the egg, we have the cytoplasm, and now we have the donor cell, the somatic cell. So the last piece, after we've had the genetic material placed in our egg cell, is to promote cell division, to get the somatic cell to fuse with the cytoplasm of the egg, and then to start mitosis and dividing, forming an individual. And that's what we see here. And the way that they promoted or prompted this to occur is through an electrical current or an electrical shock. And so then this would divide into 2 cells, into 4 cells, and so forth, until it made a solid ball of cells called a morula, and then eventually the forming cow, in this case, would be placed into a surrogate female so that her womb could support the development of this cow. But remember, this cow did not come from a sperm and an egg joining. It came from the nucleus of a body cell that was already diploid. So the end result is a clone. And on the left side, we see the clone, Liz 2, and on the right side, we see the cow that the clone was made from, Liz 1. So that concludes the presentation on Chapter 8 for Week 4. I hope that this information has been helpful for you to review both for your test and for your assignment. Please stay tuned for the next presentation on Week 4, which will be Chapter 9.